Today we are going to dive into diagnosis of Leishmaniasis. In the previous video we have looked at life cycle and pathogenesis. So today we are zeroing on laboratory diagnosis. How can I tell that someone has Leishmaniasis, which is caused either by visceral Leishmaniasis, cutaneous, or mucocutaneous? And as we begin, please we request you to subscribe if you haven't, like the video and share to many others that we may continue to grow with you. So with laboratory diagnosis, we want to know how can someone know that they have this disease. As we saw, it has some endemic areas like Africa, then America, and some parts of Asia. So we are going to see how we can diagnose visceral Leishmaniasis, which is known as Kala-Azar disease, or you can call it dum-dum fever. It causes dum-dum fever. Then later, part B, we shall look at also how we can diagnose cutaneous Leishmaniasis and mucocutaneous. And under each, we are going to look at both direct specimens, direct methods, and indirect methods that we can use in lab for diagnosing Leishmaniasis. So, with visceral, we said visceral means it affects internal organs like the liver, spleen, bone marrow, and sometimes intestines and lymph nodes. So the specimen of choice for Kala-Azar disease, we are going to see we need specimen like aspirates. We can obtain aspirates from spleen, and the spleen is the one which gives high sensitivity. It is highly sensitive, so it is preferred. But if you cannot do spleen, you can look at bone marrow and to a lesser extent the lymph nodes, the swollen lymph nodes. Here you can obtain aspirates. We can also use, sometimes we can cut a biopsy from these organs, like liver biopsy can be used. But as I have said, that spleen remains the specimen of choice in aspirates. Then we can also use, another specimen we can use is blood. We can collect venous blood, whereby this blood, we can use it as whole blood in cases of PCR, or we can use it as serum in serological tests if you want to do antibody antigen tests. Then we can also use it, we can obtain buffer coat. This buffer coat will enable us to perform thin film to examine for presence of mastigotes. And if it is, yes, for mastigotes, then whole blood we have seen we can use it for culture and PCR test. That is the specimens we are going to look at under visceral Leishmaniasis. So after knowing the specimens of choice, now let us look at the methods themselves. And we are going to begin with method number 1, which is called microscopy. And under this microscopy, we are going to obtain either aspirates or blood, we obtain buffer coat, or we are going to obtain a biopsy. Then we first make a thin film. You make a thin film or smear. You air dry the smear. After air drying it, you fix using absolute methanol. So after fixing this smear, we are going to stain the smear. And there are two methods we can use to stain them. We can either use 10% Gimusa as a staining solution or reagent, 10% Gimusa can be used. But if you don't have 10% Gimusa, we can also use H&E. That is a histopathological stain. Hematoxylin and eosin stain can also be used. And these stains will enable us to demonstrate LD bodies. These LD bodies are the intracellular mastigotes. These mastigotes are got in macrophages and monocytes when you have used the blood. So they are intracellular, located as we're going to see under the diagram. But how do we stain this thin film? With 10% Jimus after fixing, you cover the smear. If this was a thin film, so what you do, you're going to cover this thin film after fixing with the 10% Jimusa for 10 minutes. After 10 minutes, you wash off excess and leave the buffered water to differentiate. After differentiating for 2 minutes, you wash off excess, air dry when the tail is facing upwards. You dry and examine. So after staining, we are going to examine microscopically using times 40 and times 100 oil immersion objective. And we are going to look for the LD bodies, Leishmania Donovani bodies, which are known for causing visceral leishmaniasis. And how do they look like under microscope? If you examine them under microscope, let me say this is a field. This is a microscopic field under times 100. We said these are found in macrophages. So if this is a macrophage, or sometimes we find them in monocytes. If we have used the blood, when we use aspirates, we find in the macrophages. When we use blood, we find them in monocytes. So we are going to find, if this is the nucleus of a monocyte or a macrophage, we are going to find multiple LD bodies in the cytoplasm of the macrophage. If it is monocyte, still we find multiple LD bodies. This part is the nucleus. It has kidney-shaped nucleus. The nucleus is kidney-shaped. So if this is the nucleus, we find multiple LD bodies under microscope. This is the LD bodies we find. So this is how we can describe the microscopy, which was test number 1. Then number 2, we can also look for the presence of antigen, detecting presence of the Leishmania antigen. And when we are looking for antigen, those are the protein materials by the Leishmanian Donovan. We can perform PCR, polymerase chain reaction, whereby we are using DNA of the Leishman Donovani. We replicate, we amplify by increasing in the number, and we determine the number of copies. Or we can do what we call Catex. Catex, this is a latex agglutination test. So this is, we are calling it latex agglutination test, whereby the sample we use is urine. We are going to obtain urine. After obtaining this urine, we heat it for 5 minutes. Why are we heating it? To kill any presence of other proteins that might cause different reactions. So after killing those proteins, we shall remain with the antigens of the Leishmanian Donovan in urine. So what we do, it is like a rapid test kit, whereby we are using a latex part. So what we do, we come and put a drop of urine. So if this is a latex containing different sites, so what we do, this latex is incorporating antibodies. So it has antibodies which are specific to the antigens of Leishmania in urine. So when we add urine and we lock or we mix using a rotator, we are going to see, that is after adding urine, we must have a control, that is the positive and negative plus the test. So when we see agglutination on positive, there must be agglutination. On negative, no agglutination. And also on positive, we must see agglutination. This one will indicate presence of Leishmania donovani. These antibodies are commercially prepared and immobilized within the reagent. So we find them in the reagent and we put on a latex card. So this is a latex card we are using to determine presence of antigens in urine. Then another test we can do is culture. So under direct tests, the third one we can do is culture. And the culture media which is preferred for Leishmaniasis donovani is known as N media. This is the one which we are using, N media. This is the media of choice for culturing these Leishmania donovani species. So this NN media, that is McKenny-Nicole media, we culture it and we put aspirates. So if this is a culture media plate containing nutrients for Leishmania donovani, so what we do, we come and inoculate the aspirate which from a patient whom we are suspecting can be an aspirate or blood. So we aseptically inoculate it on this culture media, and then we intubate at 37 degrees centigrade for 24 to 48 hours. After that, we examine for growth. And of course, during culture, when we culture the aspirate or the blood, the amastigote which are in these samples, due to change in the temperature, are going to metamorphose into the promastigotes. So when we are after culturing, we are going to use a briquette stick. We pick some of the colonies and we make a thin film. We make a thin film. We stain it in the same way using Jimusa or H&E. Then we examine. Here we shall examine for presence of promastigotes. Note, here in direct method we are using a mastigotes, but here we are looking for promastigotes which look like this. So these are the what we call, it has nucleus at the center. Then there is what we call the kinetoplast, the axoneme, then with a small, this one does not. So this is the nucleus, then the axoneme, then we have the flagella. So this is what we call the promastigote. It is the one we are going to look when we are examining preparation from any culture media. We look for the presence of the promastigotes, not the intracellular amastigotes. So this is what we can discuss under direct methods. So let us now look at indirect methods. How can we use indirect methods like detecting presence of antibodies, that is IgG, IgM, or doing other hematological tests as we saw in pathogenesis, how it can lead to pancytopenia. So we can do those tests. And the first one we are going to do under indirect is the Fomogel. Fomogel method test. Or you can call it Napier's aldehyde test. Aldehyde test. So this is looking for presence of IgG antibodies. And how do we do it? Here we are going to look for presence of IgG antibodies, whereby we are going to collect at least 5 mL of venous blood. And after collecting these 5 mils of venous blood, we are going to centrifuge. Of course, you collect them in a red top. You leave it to clot. After clotting, you centrifuge to obtain serum. So after getting clear serum, which is free from any blood cells, you're going to get a clean tube. You add 1 ml of serum from the patient. After adding 1 ml of serum, come and add 2 drops of 40% or concentrated formaldehyde, formalin. Then after adding them, you mix. After mixing, you intubate for 30 minutes at room temperature. So after 30 minutes, but you're going to look for presence of whitening and gearing. So we examine for the whitening, the serum whitens and gels. Many commonly it gels within 5 minutes. But if it is not 5 minutes, in 5 minutes it means it is severe. Then between 10 to 20, that is moderate. Then from 20 to 30 is mild. So you examine, you keep checking within 30 minutes. So greater than 30 minutes without getting, that one becomes a negative test. But this test is non-specific. It's non-specific, so you cannot know whether someone is having a current infection or past infection, because it becomes positive after 3 months. And when you treat someone fully, it remains positive for 6 months after treatment, after recovery. So you cannot tell if someone has a current or past infection. And it is non-specific, whereby it can give false reaction in case there is also high production of IgG in cases of people with multiple myelomas and some leukemias. So it can give a false positive reaction. That's why we say it is non-specific. But before we continue with indirect methods, under direct methods, when we are making thin film, we talked about aspirates, but we can also make thin film from a buffet coat. Buffet coat, that is when we are using whole blood, whereby you put it in a small tube and you centrifuge at. So if we are using blood under direct method and we want to do microscopy, you centrifuge in a thin tube for 1000 gravity for 15 minutes. So you centrifuge at high speed for 15 minutes, then remove the plasma and obtain a white buffet coat. And it is this white buffet coat that we are going to use also to make a thin film. So not under direct microscopy, we can use aspirates from the spleen, from the lymph nodes, or from the bone marrow, or we can obtain buffet coat when we are using whole blood. That is what I wanted you to note. And how do we get a buffet coat? We aspirate this blood in a small tube. We can even use a microhematocrit centrifuge, and we centrifuge at this high speed for 15 minutes, and we obtain a white buffet coat, which we shall use to make a thin film. That is an addition I wanted to add under direct methods. So let us continue with the direct method, whereby number 1 we have seen it is the Formal test, testing for presence of increased number of IgG antibodies when someone has Leishmania donovani. Then another indirect test, which is the second one we can use in diagnosis, is RK39 test, antibody test. So this one is using recombinant gene, and the K39 antibodies undergo gene recombination to form them. So these antigens, so this is a rapid, it is acting as an RDT, and it is commonly used by researchers in resource limiting areas, whereby you can easily go with this rapid diagnostic technique, detecting having antigens which undergone recombination, detecting for presence of antibodies in the patient's serum. So if this is a rapid kit, you just prepare the blood, obtain serum in a field, add a drop of serum on this kit. You add like 50 microliters of serum. Then you time per manufacturer's manual. So a positive result indicates presence of antibodies in someone's serum, which are specific to the Leishmania antigens in the kit. So this one is being used by researchers in the field and in areas with a limited resource. We can use this as a test. Another one we can use is DAT, direct agglutination test, can also be used. This direct agglutination test, it is also looking for the presence of antibodies in the patient's serum. Here we are using serum. So in this, we are using a well which is a V-shaped well, and this V-shaped well is coated with Leishmania Donovani antigens. These antigens are be prepared from culture medias, commercially prepared. So what you do, you obtain patient serum, and this serum of the patient we are suspecting to contain immunoglobulins, which we call antibodies, which are specific to the LD antigens immobilized on the well. So when we add this serum containing the antibodies, these antibodies are going to come and bind to the coated antigens. It is a multiple well, has many wells, so we shall look for the titers, whereby the highest titer will be the agglutination, visible agglutination, which is seen least, is what we shall consider as a titer. So this is what we call DAT, looking for presence of antibodies from patient serum using multiple wells immobilized with different concentration of antigens commercially prepared. And you look for presence of a blue bottom, because we use a stain known as, which is blue in color. So you look for the formation of blue color. So if here there was the blue color, at the bottom here blue color, and the intensity continues reducing, and you find it has stopped here. This is 1, 2, 3, 4, 5. So the titer, we shall take this one, because it is the last one, which is agglutination, which is visible. That is under direct agglutination test. Lastly, under indirect, we can see is hematological tests. Under hematological tests, we are going to perform CBC and ESR. These are the hematological tests we can perform. As we said under pathogenesis, these Leishmania bodies have ability to infiltrate in the bone marrow, causing hypocellularity of the bone marrow. So we see when we do HB estimation, it might be reduced. So we are ruling out anemia. We can do platelet count, and when it is reduced, it indicates thrombocytopenia. We can do both total white BC count and differential count. And total white BC count is also going to be reduced. So presence of, here we are looking for anemia, thrombocytopenia, and leukopenia. This one can also give us a clue about Leishmania. And when we perform ESR, it is also going to be raised. ESR, that's whereby we fill whole blood from EDTA tube up to 0 millimeters mark from Westergren tube. Then we make it to stand at perpendicular angle for 1 hour. Then we see how much the rate the cells are able to sediment. And it is also raised in visceral leishmaniasis. So these are the indirect methods we can perform. That is Famogel test or aldehyde test, which is non-specific. We have seen RK39 antibody test, then direct agglutination test, and hematological tests, which are part of the indirect tests we can perform to diagnose visceral leishmaniasis. Lastly, let us look at cutaneous leishmaniasis, which causes skin ulcer, and mucocutaneous leishmaniasis, which affects the buccal cavity and the nasopharyngeal cavity, nasopharynx cavity. So here are the specimens we are going to use. We are going to use scrapings from the ulcer around the undulating or from the ulcers. We can use aspirates or lesions from the mucous membrane which are affected. These are some of the tests we can use. Or sometimes we can also use specimens like blood, but though blood is not preferred. Here we can also perform direct methods like microscopy on the aspirates, and we are demonstrating presence of amastigotes, the way we saw under Visceral. So I cannot repeat this. It still remains the same. The difference here, we are using scrapings from the ulcers or lesions from the inflamed mucous membranes. But this description remains the same, and we can stain with either Jamusa or H&E stain. And we look for the intracellular amastigotes. Apart from that, we can also do culture. We can do culture on NN media, and also we demonstrate presence of promastigotes. As we saw here, the presence of promastigotes for cutaneous and mucocutaneous. Here we are looking at Leishmania braziliensis, Leishmania tropica, major, and mexicana. They are the one we are looking for under cutaneous leishmaniasis and mucocutaneous. Then apart from these two direct methods, we can also do an indirect method. And this indirect method here we use is the leishmaniasis, leishmanin skin test, or sometimes called Montenegro reaction. So with this method, we obtain treated promastigotes or amastigotes from the culture. Then we treat them so that they cannot infect the patient. And after making this preparation from either the promastigotes or amastigotes from the culture, we are going to inject. We inject 0.1 ml of this preparation on the forearm of the patient being suspected to be having leishmaniasis. Then here we are going to observe for the swelling within 48 to 72 hours. Excuse me. So in case, because we are saying this injection contains the antigen, then the patient has the antibodies. So they are going to react to form antigen-antibody complex, which is going to trigger cell-mediated, delayed cell-mediated immunity in the patient after injecting. So this reaction will be seen within 48 to 72 hours by the presence of a swelling at the site of injection. So when we measure the swelling and it is greater than 5 millimeters, that one indicates a positive test for either cutaneous or mucocutaneous leishmaniasis. So these are the methods we can discuss with you under the diagnosis of leishmaniasis, whereby you have looked at both visceral and mucocutaneous and the direct methods and indirect methods we can use. Hope you have learnt a lot. Remember to like and share to many others. Thank you so much.